Hello, Sarah. I am going to be responding to your paper 3. Just making sure it's all there because I think that might have been an issue. Okay, cool. Should stem cells be used in research? The general definition of stem cells is that they are at the origin of a lineage and they are self-renewing. They are also able to generate all cell types of a given tissue or organism. Stem cells can be useful in many different ways. They can be utilized in a variety of different treatments, and in research, stem cells are most useful in replacing dead or dying tissue since they are able to regenerate. They are most commonly used in replacing damaged cells from different ailments such as Parkinson's disease, stroke, leukemia, and other cancers and diseases. Even though they are able to treat many different things, there is still some controversy over whether or not they should be used for treatment or in research. Okay. You're leading us in the general definition of stem cells is that they are at the origin of a lineage and they are self-renewing. I feel like you could have a first sentence that would kind of bring us in an easier way, because when that sentence is first, I'm like, the origin of a lineage? Like, just that idea, I have to stop and be like, okay, what does that mean? And then I want to hear you kind of explain it. And they're self-renewing. Stop. I want to hear you explain what that is. Okay, the two main types of stem cells are embryonic stem cells and adult stem cells. It's been so long since I've heard about this. This is cool. Adult stem cells are derived from many different places, such as the brain, bone marrow, blood vessels, skeletal muscles, blah, blah, blah. They are undifferentiated cells found among differentiated cells in a tissue or an organ. So I know this is a difficulty. Like, who's your audience really for this paper? I'm fine with you writing it like more to medical professionals, but I feel like you could explain quickly what an undifferentiated cell and a differentiated cell is. I think I know what that means, but it also might be just helpful for your understanding to go ahead and try and put in a little definition. Because when we have to explain something to other people, sometimes I feel like always we learn more about it. Most of the stem cells that come from embryos that develop from eggs that are fertilized. Most of the stem cells come from, you mean embryonic stem cells that are used in research. Not most stem cells come from them, I think. Fertilized in vitro, in other words, fertilized in lab setting. These cells are grown by placing them in a culture medium in addition to allow them to grow. They used to be developed using mouse feeder cells, but researchers have now found a new way to develop them without it since there was a risk of transferring mice viruses to the human cells. Well, shoot, that's the last thing we need. Scientists first discovered that stem cells could be used in 1981. Scientists were able to derive embryonic stem cells from mouse embryos, which paved the way to future stem cell research. So those are probably examples of things that are such accepted facts that you probably don't need to cite them at all. Again, I would say, like, it's rare that you're going to get in trouble for citing too much. Sorry, but in these cases, like, it breaks up your writing a lot, and I'm like, well, everyone on the planet must agree that, I don't know, they may very well agree that it happened in 81 and that they came from mouse embryos. And if that's the case, you don't really need to cite it. In 1998, this led to two scientists discovering how to derive stem cells from human embryos and grow the cells in a laboratory. In 2006, researchers made another breakthrough. They discovered that already specialized adult stem cells could be placed in certain conditions and be reprogrammed to revert back to a stem cell-like state. This is also called something, and that's great. These are both advantages and disadvantages. There are both advantages and disadvantages using embryonic stem cells and adult stem cells. The advantage of embryonic stem cells is that they are able to be easily grown. We don't know what adult stem cells are yet, though, right? So maybe we should find about that before we hear about comparing them. The advantages and disadvantages. They're likely to be rejected by a patient since they are not their own cells. The advantage of adult stem cells is that they are less likely to be rejected since they are the patient's own cells. Now I'm confused, though, about, like, how do they harvest adult stem cells? Is that like bone marrow transplant? Or the main disadvantage is that they're thought to be limited to differentiating to different cell types. This is because since they come from the tissues and organs that are already specialized, it is more difficult to make them differentiate into a different type of cell. So that's still in yellow, which is making me wonder if you were going to put quotation marks around it. You forgot, so you can fix that. There may not be a clear answer as to which type of stem cell is better, but they are both certainly beneficial to different types of treatments and research. There was a study conducted by this person that showed research how to restore fertility in mice. These mice had a C something, which was a major component in the blood-testis barrier. When an animal has a deficiency, they lack the BTB, which results in a lack of spermatogenesis. Without spermatogenesis, the animal is infertile. Researchers use spermatogen, although that is meant to explore the role of certain proteins that result in deficiencies. As a result, it was discovered that some forms of male infertility could be rescued. Rescued? Maybe that's just what they say. I don't know. By this, the stem cell transplant allowed development of fertile spacejet mice. Cool. A different study conducted by these other people and others explored using amniotic fluid stem cells to repair sciatic nerve injuries. Oh, those are painful. The goal of this study is to replace the current standard for repair of peripheral nerve injury. Even though it's considered best to repair nerve damage, only about 40-50% make a full recovery. In order to conduct this study, so even though even though the current standard treatment is considered. I need to specify that. In order to conduct the study, researchers combined human with in order to repair nerve cell injuries in mini pigs. Mini pigs were used because they most closely resemble the challenges of human nerve grafting. As a result, it was found that combining amniotic fluid stem cells with conduits could expedite the repair of sciatic nerve injury. The stem cells were able to produce growth factors, which helped great potential for treating nerve injuries. They were able to facilitate nerve regeneration and lead researchers to believe this may be more practical than the other thing. Even though there are many benefits to using stem cells, there are still some controversy surrounding it. Okay. The controversy stems back to the 1990s. Back then, embryonic stem cells were derived from actual human embryos rather than from a lab setting like the way it is today. Oh, wow. Yeah, I did not know. Many people disagree with the use of embryonic stem cells because using them means destroying the embryo. Another controversy surrounding this is whether a modeled part of a human embryo should have a legal status similar to that of a completed embryo. A modeled part? As of now, the tissues and stem cells are not deemed to be biologically equivalent to a complete human embryo. I would like a little bit more about what is a modeled part of a human embryo. This leads to a question: should stem cells be used in research and for treatments? I mean, it doesn't really lead to that question, does it? I think you can just stop there and go to the next paragraph. I believe that since there are so many benefits to using stem cells, because by the fact you're talking about benefits kind of pulls us down, they should be used for trauma and treatments and research. But in order to do that, alternative solutions need to be put in place. Alternative harvesting solutions, I think is what you mean. An alternative solution to using embryonic stem cells would be to use induced something. These cells are made by making the adult stem cells from multiple body tissues go back to an embryonic-like state, which allows the cells to develop into other cell types. Since this uses adult stem cells rather than embryonic stem cells, this gets rid of controversy. They're made to resemble embryonic stem cells without actually being them. Okay, so yeah, organizational problem here that I was not really able to process and kind of consider the benefits of the one without knowing what the other one even was. So I think it makes more sense to move up this example of the other way to do it so we can compare earlier. One study conducted by Blah has researched how reprogrammed stem cells could mend a heart. Researchers in Japan were able to make iPS cells and graft them onto damaged human hearts. As a result, they were able to regenerate the damaged heart cells and prevent further damage. Wow. The cells were able to resort back to an embryonic state and replace damaged tissues. Even though the embryonic stem cells were not used in the experiment, the cells were still able to behave like them. Using alternative methods to provide embryonic stem cells using alternative methods are useful. I think you mean using alternative methods to embryonic stem cells is useful and can reduce ethical concerns. Not only can this be useful in treating infertility, it can also recreate different organs by taking these cells and conditioning them to develop into a certain organ cells. They could generate structures that resemble actual human organs, like when they grew that ear. This could pave the way for researchers to develop new organs for research organ transplantation. They're also very useful in preventing different diseases. For example, one example for where this is true is in preventing diseases. During the first few weeks of pregnancy, especially those caused by use of alcohol and drugs, subtle cell abnormalities within the first few weeks of pregnancy can cause damage to the baby. By using these cells to alter the development of the placenta, it can make the baby less vulnerable to that's really cool. I don't understand how that works. And you might say a little more, but that would be really cool. There was a study involving these conducted by a lot of research in Japan, it looks like, that looked at the possibility of restoring spatial memory dysfunction in mice. Is there a reason why they particularly studied this in Japan? Again, I'm just guessing it's Japan. It seems like a lot of names look Japanese to me. Research looked at mouse models that had cognitive deficits, senile plaque, and other forms of human dementia. In order to conduct this study, researchers substituted from groups of mice. In one group, they transplanted neuronal precursors derived from iPS cells into mice. The other group acted as control groups. They did not receive neuronal precursors. Once they implanted the neuronal precursors, the researchers waited 8 weeks. After 8 weeks, the researchers found a significant improvement in the mice that had neuronal precursors, but no improvement in the mice within the neuronal precursors. This led the researchers to inquire that if this could be helpful in improving dementia in mice, that's a potential for improving dementia in humans. Although I do believe iPS cells are a good solution, I also think there needs to be a lot of regulation in order for it to be used widely. Since they closely resemble embryonic stem cells, there is some concern over what their legal size should be. Many people wonder whether they should be treated the same as embryonic stem cells or not. In my opinion, there needs to be a limit as to how closely these cells can resemble embryonic stem cells. If they're too close, that's something they're able to control, then it would reopen ethical concerns over their research and development. If there's a limit to how closely they can resemble each other, then it would reduce the concern over whether they should be treated as stem cells or not. I believe that stem cells should be used in treatments and research because they provide a number of different benefits. They can help in a treatment short of different ailments, diseases, and even some cancers. Since stem cells remain a big topic of debate, I believe that using iPS cells would be a good alternative. They're able to produce the same outcomes as regular embryonic stem cells, but without the embryos. Always, or sometimes, or we're still finding out how much. Stem cells should continue to be used in research and treatments, but alternative solutions should be considered in order to reduce concerns. So the one thing, then I'll look at your works cited real quick, just on first glance. That looks good. I think you need to do a little bit of explaining what the ethical, maybe it's really clear to everybody, but I think just a little bit about what are the ethical concerns about using, about destroying embryos, because there are people who don't have that concern at all. So just a little bit about that, I think. And then I gave you some ideas about organization. I hope you find that helpful. I found that educational. So thanks for your paper, and I look forward to seeing the revision.